Okay, Chapter 8, Section 4. Determinants of cell morphology. Why are bacilli bacilli? Why are cocci cocci? Why are the rods rods? The sphere sphere, and everything in between. Well, it turns out it's all driven by the cytoskeleton. Kind of figured that. Cytoskeleton proteins are all gene products. So which genes are there will determine which cytoskeleton components are there, how they're formed. We call this SEDS. SEDS, shape, elongation, division, and sporulation. These are proteins that will determine shape, determine what the cytoskeleton looks like. There are homologous proteins, genes and proteins, in eukaryotic cells, which will, determine what our cytoskeletons look like. First one we're going to be talking about is the protein MREB. MREB, found in bacteria and a few archaea. This protein forms a simple cytoskeleton inside the cytoplasmic membrane. Turns out the cytoskeleton of prokaryotic cells, specifically bacteria, is nowhere as extensive as what you're going to find in an animal cell. So that cytoskeleton that you learned about, you studied, talked about in protist and animal cells that you discussed in cell biology, intro cell and molecular cell biology, you're not going to see anything quite that as advanced here. What you're going to have is a patch-like filaments. Okay, so certain areas are going to be heavy in it, certain areas are going to be lacking in it. The MREB protein is going to recruit proteins to form what are called an elongisome. A longisome. Its job is to elongate the cell. Makes sense. MREB, when active in cells, causes them to look bacilli. Inactive causes them to foreshorten and looks like cocci. It's one of the things we ran into in the lab. Some of the students who were, gram staining, their E. coli didn't look quite as elongated rod-like as other people's. Theirs looked shorter, rounder, more elliptical, where other people had those nice elongated. And I told them, oh, it's just something about the growth characteristics. They got starved or something like that. In actuality, what it is their MREB proteins were inactivated and those genes weren't expressed. Because you inactivate them, what should be a bacilli will start to become looking like cocci with each successive generation. Now, things change, boom, nutrients come back, whatever. MREB will become activated again, that gene will be overexpressed again, and all those resulting daughter cells will start looking more and more like bacilli once again. Bacteria that are supposed to be coccus, Staphylococcus aureus, Streptococcus pyogenes, things like that, completely lack MREB and ROD-Z. These are the two proteins that are responsible for this filamentous, cytoskeleton, which causes the elongation of the cell and be looking bacilli. So these elongisome complexes, you'll see stretched throughout the cell. Their job is to make the cells stretch. Stretch and pull. Boom, boom, boom. Stretch the peptidoglycan layer. Stretch the membranes. Certain prokaryotes, instead of using those proteins, they're going to use a protein known as crescentin because they're going to take on more of a crescent shape. We see this in Colobacter. We see this in Helicobacter pylori. Remember, Colobacter is the one that looks kind of like a bean with a stalk because it's sessile. Helicobacter pylori is a flagellated protein or flagellated bacteria that you're going to find mostly in your stomach. Both of these have a somewhat comma or kidney bean shape to them. So what we saw on the previous slide with the gram-negative and the elongisomes, not going to work here. MREB, okay, it's going to work with microfilaments, actin, microfilaments, forming a scaffold of sort. Yes, you're going to see FITZ also coming into play here. It's important, the FITZ, which can interact here, it's going to be important in the cellular division, but the MREB, its job is to elongate, which is important, especially if you have E. coli that's going to be cutting itself in half. I'm not going to talk about those right there. So it's interesting. Up till now, we've been talking things, referring to bacilli and talking about it being in the middle, kind of like E. coli does. But we have to keep in mind E. coli is not the only bacteria out there. You look here, Agrobacter tumefaciens. Tumefaciens, its growth is one of the terminal ends of the bacteria will begin to elongate out. The glowing green here, this is where you have the proteins and things that are associated with elongisomes. And this is where you're going to see new cell wall synthesis and things like that as the cell begins to push out. Here, 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 boom. So we see this pushing out here. New cell being made. Any new chromosomes are pushed in there. And then right here, you have all of that FITZ stuff that we talked about last section.